I'm Murtuza from EMD Serono, and I head up the Oncology Marketed Products Global Evidence and Value Development Group.
I'm Venediktos, a principal statistician from Evidera.
Today we will give a summary of our recently published paper, Assessment Schedule Matching in Unanchored Indirect Treatment Comparisons of Progression-Free Survival in Cancer Studies, that was recently published in the journal Pharmacoeconomics. On this project, I led the study team carrying out an indirect treatment comparisons of progression-free survival and overall survival for one of our oncology products. And in the course of these analyses, we realized that our comparisons of PFS may be biased because of treatments we are comparing had used a different tumor assessment schedule than ours. So beyond population differences, which traditional indirect treatment comparison methods are able to address, there is no method to systematically address these differences in assessment schedules across studies. As a result, we sought to come up with a way of addressing this specific issue. My colleague Venediktos drove the methodological effort that led to this method, which we are calling assessment schedule matching.
Assessment schedule matching is a method that we have proposed to remove or at least reduce the effects of assessment time bias. We will describe assessment schedule matching through an example.
Presume two treatments have the same underlying PFS risk reflected by the green curve. In trials, scheduled tumor assessments for treatment 1 were every 6 weeks, while for treatment 2, our comparator of interest, assessments were scheduled every 8 weeks. The Kaplan-Meier curves for PFS derived from these trials, which give us the proportion of patients who are still alive and progression-free, will appear to differ even though they arise from the same underlying risks. As you can see, the Kaplan-Meier curve for treatment 1 in blue will drop earlier than for treatment 2 in red, implying a higher risk of progression or death. An indirect treatment comparison of these two treatments would provide biased results due to the difference in schedules, even if we were able to successfully match all the differences in patient characteristics in an MEIC. The bias would favor treatment 2 because it assessed progression later. The ASM method aims to adjust the timing of when PFS events for treatment 1 would be detected if the study had followed the schedule used in the comparator study. Here we are assuming that we have patient-level data for treatment 1, which allows us to adjust the timing of events. We do not need patient-level data for treatment 2, only the published curve and details on the assessment schedule. The ASM method for matching the first scheduled assessment involves 3 steps. A forward shift, where we are advancing the times of individual disease progression, in this example by 2 weeks, that is from 6 to 8 weeks, because these events would have only been detected at that time. In other words, all patients who were flagged as progressed at week 6 would get moved to week 8, because this is when the study visit would have occurred. It is possible that some patients are found to have progressed at an unplanned study visit. We do not move the timings of these patients, as we assume the events that triggered the unplanned visit would have still occurred and led to detection of the progression. The second step involves a correction for death or censoring. If the shifted time from the first step exceeds the time of recorded death or censoring, it is assumed that the progression event would be missed at 8 weeks. The third and last step of the ASM method involves a backward shift of recorded progression events. In our example, the second assessment for treatment 1 would occur at week 12. Some proportion of the progressions identified at this time occurred between week 6 and 8, and therefore these progressions would have been detected at a visit at 8 weeks had there been an assessment at that time. In our paper, we describe several ways of estimating this proportion, including a progression probability calculation approach based on a model fitted to the data adjusting for interval censoring, and a worst-case scenario approach that is useful in limiting the uncertainty associated with this proportion.
To assess the statistical properties of the ASM method, we conducted a simulation study based on the PFS survival function estimated in a real study of patients receiving second-line treatment for locally advanced or metastatic urothelial carcinoma.
We simulated data to mimic the way a PFS curve might look in real trials. Specifically, we wanted to ensure that the simulation could generate curves with a sharp drop at the first assessment time, as this is often seen in immuno-oncology therapies, but also cover cases with a slower rate of progression. We simulated PFS curve under different scenarios, varying parameters like the rate of early progression, sample size, and duration of follow-up. Then we created a comparator curve by applying a hazard ratio, the true effect we're trying to replicate in the analysis. Finally, we implemented different assessment schedules in each group to induce assessment time bias and compared our estimates of the hazard ratio with and without ASM.
As expected, we found that without ASM, the hazard ratios were consistently underestimated across simulations. The size of the bias was generally consistent when the sample size, hazard ratio, duration of follow-up, and assessment schedules were varied.
The most influential factor was the proportion of patients who progress or die by the first assessment time. We found that the bias gets larger as the proportion increases. Specifically, the bias was minus 11.3% on average, when around 40% were event-free at the first visit, and almost negligible, minus 2.3%, when around 80% were event-free. In these cases, such as where a large proportion of patients have a PFS event by the first visit, even relatively small differences in schedules, for example 2 weeks, may be sufficient to bias estimates of relative effect and generate potentially misleading conclusions. ASM was effective in removing the bias or substantially reducing it to the point of being ignorable across all scenarios. We also found that adjusting the first visit only was sufficient to achieve reliable estimates. That is because when a large proportion of the PFS events occur at the first visit, ASM on the first visit leaves relatively fewer events to be affected by the discrepancies in the timing of later visits.
As you see, ASM is a new method that is simple to implement to correct for potential bias due to differences in tumor assessment schedules when comparing PFS estimates from single-arm studies. ASM should be used prior to conducting any unanchored comparisons of treatments such as MAICs or simulated treatment comparisons, STCs. The method provides a what-if scenario, avoiding unverifiable assumptions, and enables unbiased indirect treatment comparisons when comparing single-arm studies. Furthermore, a worst-case scenario can easily be investigated, limiting the uncertainty around the very few assumptions made and the potential criticism on the use of ASM method in the context of HTAs. We hope other researchers consider this new method when relevant in addition to MAICs, which only adjust for differences in population characteristics.
We have also made the R code to implement this method available as supplementary material with the original article for researchers to use. As next steps, we plan to implement this method in a real-world situation and show the impact of using this method, which we are hoping to publish soon.